Hello everyone, welcome to today's live broadcast, Exosome Biogenesis and the Budding of Proteins and Viruses. I am Dr. Chad Schwartz, Senior Application Scientist at Beckman Coulter, and I'll be your moderator for today's event. We are delighted to bring you this educational webinar presented by LabRoots and sponsored by Beckman Coulter. Beckman Coulter is a leader in centrifugation and flow cytometry and has long been an innovator in particle characterization, lab automation, and genomics. Beckman Coulter products are used at the forefront of important areas of investigation and discovery. For more information, please visit beckmancoulter.com. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the icon in the lower right-hand corner of the slide window. If you have any technical problems viewing this presentation, please click on the support button at the top right of your presentation window or submit your problem through the green Q&A button. I would now like to introduce today's speaker, Dr. Stephen Gould. Dr. Gould is a professor of biological chemistry at Johns Hopkins University in Baltimore, Maryland. Dr. Gould received his PhD from the University of California at San Diego, where he studied the molecular mechanisms of peroxisome biogenesis. In 1991, he moved to an independent position at the Kennedy Krieger Institute and the Johns Hopkins University, where he continued his studies of peroxisome biology and peroxisomal diseases. In 2003, Dr. Gould began his foray into exosome biology and HIV budding by co-authoring the Trojan exosome hypothesis with his collaborator, Dr. James Hildreth. Dr. Gould's research has since focused on the molecular mechanisms of intercellular vesicle traffic, retrovirus budding, and the translational applications of these processes. Dr. Gould also serves as the president of the American Society for Exosomes and Microvesicles. Before we get started, I would like to ask a few poll questions to our audience. Our first poll question is, what separation technique are you currently using to isolate exosomes? Ultracentrifugation or differential gradient, affinity capture, size exclusion, or precipitation reagents? Please select one of the four answers, and we'll give a bit of time for the audience to respond, and I will then disseminate the results. All right, our results are in. It looks like 60% of folks in attendance use ultracentrifugation or differential gradients. 20% use affinity capture. 0% are at size exclusion chromatography, and 20% are at precipitation reagents. Our second and final question will be populating here shortly. What is your sample source for extracting or isolating exosomes? Tissues, cell culture, blood, urine, plasma, or other. Exosomes come from a variety of sources. Please just select the one that you are using in your research. A few more moments as the audience responds. And an overwhelming majority use cell culture at 83.3%, and 16% use plasma. I would like to thank everyone for their answers, and I will now turn it over to Dr. Stephen Gould for his presentation.
Good morning. Thanks, Chad. Thanks for having me. I'm just trying to clear out some of this stuff on the screen. Okay, so yeah, I'm Stephen Jay Gould. Welcome, and I'm in the Department of Biological Chemistry at Johns Hopkins University. And I want to speak to you today about exosome biogenesis and protein budding and virus budding. Before I get into that, though, I just want to make sure everybody's aware of the upcoming meeting of the American Society for Exosomes and Microvesicles. ASEM 2015 is going to be held in late October, beautiful resort island, Marco Island in Florida. We're having approximately 200 attendees, and we're happy to announce this year that Beckman Coulter is our primary sponsor for this event. So if, please register for the meeting, and you can do so online at asemb.org, and I look forward to seeing you all in Florida. Now, just a little bit of background before we get into the meat of the talk. And what are exosomes? What are we really talking about? Well, so they are, in my opinion, secreted vesicles, have a variable diameter depending upon the cell type in which they're derived from, around 50 to 250 nanometers. They have the same topology of the cell. They really have no other defining physicochemical properties, and they're released by all animal cells tested. Now, the term exosome was originally coined by Trams et al., and in their paper, they define exosome as secreted vesicles that, quote, may serve a physiological function. I think this is a very reasonable definition. Now, roles of exosomes in normal physiology are quite varied, and I like to think of them in terms of whether they are playing a role for the vesicle-producing cell or whether they're playing a role in signaling to other cells. And you can describe these as cell-autonomous effects or cell-non-autonomous effects. And in the cell-autonomous effects, there's good evidence that they're playing roles in protein quality control, in cell polarity, differentiation, and modulation of the extracellular matrix. And of course, the great interest in this field now is in the cell non-autonomous effects and the intercellular transfer of signals, molecules, and genetic information. A great example of the role of exosomes in protein quality control was published just last year by Gavin Wright and colleagues, in which they were studying the protein Juno, which is the sperm receptor on the surface of mammalian eggs. And so in the upper panel of this, upper left panel of this image, you can see prior to fertilization, immunostaining for the Juno protein is quite bright at the surface of the egg. And after fertilization, the protein is rapidly lost from the cell surface. And when Dr. Wright workers looked at what was going on, what they found is that the protein just was budded straight off the surface of the cell. And this is shown in the bottom panels. Here we have the egg prior to fertilization, and you can see the immunogold labeling for the Juno protein right at the cell surface. But then shortly after fertilization, it's absent from the cell surface. You no longer see immunostaining here, and instead, the immunostaining is present in vesicles just off the surface of the egg. Just a beautiful demonstration of the role of exosomes in modulating the protein content at the plasma membrane. As I mentioned, most current interest in exosomes are as intercellular vesicle trafficking vehicles. And this is a beautiful cartoon in which one can see the exosomes interacting with cells in various ways by engaging cell surface receptors, perhaps fusing at the cell surface, being endocytosed, perhaps fusing after endocytosis, with the net effect that information, both signaling information and actual transfer of molecules and genetic information, can be mediated by this pathway. Now, intercellular vesicle trafficking has been implicated in immune signaling, in germ cell maturation, Wnt signaling, angiogenesis, tissue repair, transdifferentiation in stem cell biology, and many other processes. They also play very important roles in pathophysiological situations. In cancer, we know that there are cell autonomous effects. Vesicular secretion of anti-cancer drugs, particularly doxorubicin, has been reported. Engaging this pathway, putting more cargoes into this pathway, appears to induce amoeboid morphology, and amoeboid morphology in cancer is highly correlated with aggressive metastatic cancers, and also degradation of extracellular matrix demonstrated by a number of labs. But again, also vesicles released by cancer cells have been shown to contribute to the bystander effect, whereby cancer cells alter the physiology and gene expression profiles of the neighboring stromal cells. Also in neurodegenerative diseases, where they're involved in the transmission of the pathology, in the case of prion diseases, actually transmission of the infectious protein. And of particular interest in our lab involved in viral biogenesis, HIV and other retroviruses, but also hepatitis C virus, and even non-envelope viruses like the picornaviruses, poliovirus, Coxsackie, hepatitis A virus. These actually are emitting from cells within vesicles and can be taken up in that form as well and be infectious. So they're very important. And the importance of the vesicles is reflected in the fact that they have been recognized by NIH as a topic to be studied under the NIH Common Fund, and particularly the Extracellular RNA Communication Consortium. And I just show you here the link to their really beautiful web show about extracellular RNA and the role of exosomes in transferring that out of the cell and into other cells. Now, a little bit about how exosomes are purified. As Chad mentioned, differential centrifugation is commonly used. It's what we use almost exclusively in our lab, and it still remains the gold standard. But there are other techniques for purifying exosomes: polymer-induced precipitation, filtration, either membrane or gel, affinity purification using lectins, antibodies to specific markers have all been used. But the technique that we use in our lab and that we use again and again for the types of experiments I'm going to be talking about today is outlined here, where we simply remove cells by a low-speed spin, pass the supernatant through a 0.2 micron filter, do a couple of 30-minute spins at 10 kg, in which we transfer the supernatant over then for high-speed ultracentrifugation for an hour. And we arrive with a population of vesicles that are shown in this electron micrograph. These are exosomes released by human T cells in culture, very uniform size of about 50 to 80 nanometers in diameter, and a fairly indistinct morphology. Now, exosomes have been considered together with many other types of extracellular vesicles, and various different names have been coined to discuss extracellular vesicles: exosomes, exosome-like vesicles, microvesicles, microparticles. And one can find cartoons like this. This is from AbCam, but in many reviews in which characteristics and definitions are offered in terms of the types of proteins that are found, their sizes, their densities, etc. And I would just simply comment that there's really not an awful lot of evidence to support these definitions. And another weakness of many of these is that it really does almost always emit viruses and virus-like particles, even though many cells, in particular cancer cells, are producing large amounts of virus-like particles. So one can think of the extracellular vesicle heterogeneity issue as having a spectrum of views. On one extreme, you could have the splitters view, where really all of these variations in vesicle size and composition are reflecting independent mechanisms of vesicle biogenesis, each mechanism generating a distinct type of vesicle that has distinct non-overlapping sets of cargo molecules. And so the splitters dream of the field. And you can go to the other end of the extreme and have the lumpers view, that the heterogeneity really isn't reflecting multiple mechanisms of biogenesis, but rather just stochastic variation in a single primary mechanism. And the reality is that it probably lies somewhere in between these two, but the truth of the matter is right now we don't really know, because we don't know much about the actual mechanisms of vesicle biogenesis. Now, what I'm going to talk about today is not a lot of general models, but rather actual primary data. And I want to draw your attention to the people who actually did the work. Francis Fortura, who's a current graduate student in my lab, but also I've been blessed by having a number of really great students work for me over the last few years. Zhiming Yang, Xin Gan, Ning Wu, Beiyi Shen, Amy Booth, have all made major contributions in my lab to trying to understand how vesicles are made, how proteins bud, and the relationship between that process and how viruses bud from cells. And I do want to mention James Hildreth, Dr. Hildreth, professor of pharmacology here at Hopkins for many years, who's then now gone on to take his research first out to Meharry Medical College, and then out to UC Davis, where he's served as Dean of Biological Sciences, and I believe he's now back at Meharry this year as a Dean of the Medical School. So these people have made major contributions to what I'm going to be discussing. Now, one way to think about the work we do in my lab is that we take a cargo-based approach to studying exosome biogenesis. Rather than focusing on a vesicle or this type of vesicle or that type of vesicle, we focus really on a protein or a set of proteins. And I'm going to talk about just a small number of them today. And our goal in these studies is to identify and characterize the cis-actin targeting information in these proteins. What are the parts of the protein? What are those elements that really are sufficient to get the protein to sites of vesicle budding and out of the cell in exosomes? Ideally, down the road, we'd like to identify and characterize the transacting factors, the gene products and genes in the cell that are involved in that process of recognizing exosomal cargo proteins, delivering them to sites of budding, and making the vesicles and getting those cargoes into them. And then based on the data that we obtain from these kinds of studies, obviously our goal is to develop and test mechanistic models of exosome biogenesis. And I think this general pathway will be successful in this field. It's the general approach that's been used in cell biology for several decades now to elucidate pathways of organelle biogenesis and for organelles that exist inside the cell. So what is the current view of exosome biogenesis? Most people view exosomes as vesicles that are only made by budding into endosomes and then released only after endosomes fuse with the plasma membrane. And that sort of view is shown in this cartoon on the right, where you have vesicle budding, outward budding domains, and a limiting membrane of the endosome. These bud and pinch off to form intraluminal vesicles, which then accumulate. We get a multivesicular body forming, and then that moves to the plasma membrane, docks, engages, fuses, and releases exosomes in a pulsatile manner. Now, when we started working in this field, we started looking at human T cells for reasons that will become apparent later, and other leukocytes. And when we stain these cells for using antibodies to detect exosomal cargo proteins, such as CD63 shown in the top panel, CD81 shown in the top panel, or looking at what were known to be exosomal lipid cargoes, so modified forms of phosphatidylethanolamine, PE, are trafficked by cells to sites of exosome budding. And you can see on the left of both of these images the concentration of these fluorescent forms of PE at distinct domains of the plasma membrane. So here's the phase image to the right for these, and here we have our exosomal cargo proteins as well. And what you see actually is that the proteins are accumulating at discrete domains of the plasma membrane. And when we saw this, we were obviously a bit surprised to find these exosomal cargoes at the cell surface, although perhaps we shouldn't have been, given that these proteins are actually CD proteins, which means that they were defined by their existence at the plasma membrane. But we found them at the plasma membrane. We found them in discrete domains. And Yi Feng, a student who was doing some of this work at the time, along with Amy Booth, realized that this had been seen before. It might give us clues as to what's going on. Furthermore, what we showed is that these proteins were not merely at the cell surface, but they were budding directly from the cell surface. And you can see that here with CD81 containing vesicle budding straight from the plasma membrane, and also again in this image as well. Now, as I mentioned, this is reminiscent of other observations in the field of cell biology, and particularly the capping and shedding of cross-linked cell surface proteins. And this actually was a fairly intensive area investigation in the late 60s and early 70s. Marty Raff, Inanue, and others did a lot of hallmark studies in this area. And essentially what they showed is that if you had a protein like CD43 on a lymphocyte, and you stained it, stained the living cells, you'd see that it was all over the cell surface. But now if you added a cross-linking secondary antibody, what happened is that rather quickly, the proteins came to exist in just a single pole of the plasma membrane. This was thought to be driven by the binding energy of mutually multivalent antibody-antigen reactions. It's allowed by the fact that you have a fluid plasma membrane, which allows diffusion, lateral diffusion in the plasma membrane. And you get these caps forming a single pole. And it was shown in a small number of studies that these cap proteins are subsequently released on membrane vesicles. It didn't really attract a lot of attention, this latter part. But what came to our minds was that this could actually be the targeting information of proteins, i.e. just simple aggregation or high order oligomerization might be what's driving proteins to bud from cells. So following up on that, experiments were done in which you labeled a T cell with antibodies to CD43, which does not normally bud from human T cells. And we also labeled them with RHPE, and you can see that here. It defines the cap of where exosomes are being made in these cells. And you can see it's just a small domain of the plasma membrane. Now, if you take these cells and you add a secondary antibody to cross-link the CD43, it goes to these caps. And what you can see here is that the number of vesicles that are now labeled green goes up a couple orders of magnitude. And by immunoblot, we actually don't get anywhere near as robust in increasing the budding, but you can see it also here, that after the cross-linking of the antibody, you can see it come off in exosomes, which is the upper panel, with the cell-associated levels in the bottom. So high-order oligomerization is targeting plasma membrane proteins to sites of budding and into exosomes themselves. And we showed this for a large number of cell surface proteins. So it's not that the cargo proteins in these instances have a classical type of targeting signal. They really don't normally go to exosomes, but you can get them to go to exosomes simply by adding a primary antibody and a cross-linking secondary antibody to cause essentially their aggregation in the plasma membrane. Now, we also asked, was the corollary were true? If we took a protein that we know forms high-order oligomers in the cytoplasm but doesn't normally bud from cells, can we get that to bud by putting it into a cellular membrane? So for this, we turned to a protein called TYA. It's actually a yeast protein. It's part of the TY1 LTR retrotransposon in yeast. And this is a non-viral element. It never buds from cells. If you express it in mammalian cells, as shown in the upper left here, you see that it just piles up in the cytoplasm of a human T cell. And again, it does not bud. And this is our site of budding. Okay, so that does have these plasma membrane domains in this particular cell, enriched for the exosomal target protein. But now if we simply add 10 amino acids to the N-terminus of this protein, we find that now it not only goes to the plasma membrane, but it goes to these caps, where it co-localizes with exosomal marker proteins. And if you look at how these proteins bud, here the upper panel of the exosome fractions and the bottom panel is the cell-associated fraction, you can see that the TYA protein, we fused these proteins to GFP, by the way, that it's expressed quite well in these cells, but it does not bud from cells. So nothing buds out of these cells. But if you now take this protein that has this 10 amino acid tag at the N-terminus, and this tag specifies myristylation at this glycine position right here. The protein now is expressed well and buds well from cells, which you can see quite well here. It actually buds from cells just as well as HIV GAG, which is a viral capsid protein that mediates the budding of HIV. If you mutate that glycine residue, the protein no longer associates with the membrane, no longer buds from the cells. So basically, adding a plasma membrane induces the budding of this high-order oligomeric complex protein, TYA, from cells. Now, if one looks at the exosomes one derives from these cells, it's kind of interesting. I mentioned before that these particular cells make exosomes of a very uniform size, about 50 to 80 nanometers. But when we express this one protein, acyl-TYA, we find that it actually accumulates quite well in the secreted vesicles, which you can see by the arrows in the slide. They're pointing not to the membrane, but to an electron-dense lamina just underneath the membrane that's composed of the TYA protein. And not only does it change the morphology of these exosomes to the point where they now more resemble a virus-like particle, but the sizes and shapes and densities of the vesicles change also, just by expression of one protein cargo. The sizes tend to be larger. They go up to the 200 nanometer, 300 nanometer range. They tend to be very irregular. They have tails on them sometimes, and the density is much higher than normal. So properties like size, shape, density are determined by the cargo proteins that are put into the vesicle, and simply expressing one protein in a cell can change these dramatically. Now, we've looked at other plasma membrane anchors to see how they function in terms of being able to induce the budding of TYA and other high-order oligomeric proteins. And what we find is that there's a large number of plasma membrane anchors that can do the job. I mentioned previously that myristylation tag, and again, this is a 10 amino acid sequence, but really all that's essential is this glycine at position 2 of the protein. If that's a glycine, then it's quite likely that it will be recognized by the myristoyl transferase, and the protein will be acylated at that position. You can put at the C-terminus prenylation palmitylation tag, and that will also be sufficient to induce the budding of TYA. At the N-terminus, if we put in a PIP3 binding domain, so this is from the protein ACT, it binds to phosphatidylinositol 3,4,5 trisphosphate, which is present at the inner leaflet of the plasma membrane. That's sufficient to induce the budding. A PIP2 binding domain, it can also be done. And again, what I want to point out is you can anchor this protein in the plasma membrane at the N-terminus and at C-terminus by acylation, by a lipid binding domain, and any of them are sufficient to get the protein to bud from cells in vesicles. And in fact, I mentioned to you earlier that protein CD43 that does not normally bud from cells, but can be induced to bud by adding extracellular cross-linking antibodies. Well, you can also get that protein to bud by adding a high-order oligomeric protein to its C-terminus. If you fuse CD43 to the TYA protein, now you find that it goes to sites of vesicle budding and buds from the cell in exosomes. Now, what these data show us is that when we're asking the question of where these types of high-order oligomeric complexes bud from cells, it appears to be that it's the plasma membrane anchors that do a very good job of inducing their budding. But what about if we target these same cargome proteins like TYA? If we use a different type of lipid binding domain and we target them to the endosome membrane, what happens then? Well, that's shown in this slide. What we've done is we've fused TYA to a tandem duplication of the PI3 binding FIVI domain. So this FIVI domain here binds to phosphatidylinositol 3-phosphate, which is an abundant lipid in the cytoplasmic base of the endosome membrane. And what we find is that the targets the TYA GFP protein to these internal organelles. They do not obviously co-localize with the exosomal cargo lipid here, although they do co-localize with an endosomal marker, which you can see in these lower panel. And if we ask how well it buds, not too well. So there's a comparison of the budding of the acyl-TYA GFP protein, so where we put the TYA in the plasma membrane by virtue of that meristylation site. It buds very well from the cell, and that's reflected here in the actual quantification from multiple trials. You'll notice that the budding of the endosomally targeted form is not 0. It's about 1%. And there actually is a band here if you look carefully enough. Oops, sorry, I messed that up. Reverse that arrow, it points to a band. So it's not that it's inconsistent with budding, it's just that it's not very efficient at inducing the budding of proteins. And the confirmation that exosomes can bud from the plasma membrane of these cells is shown here, where we're looking at cells that are expressing that acyl-2IA protein, and on which CD43 has been cross-linked with antibodies. In this top panel, you can see that all these cargos are co-localizing at the discrete domains of the plasma membrane. Is the merged image. And at the ultrastructural level, what you see is the cell surface of largely devoid of budding profiles, except at one patch of the cell, just one pole of the cell, where you see all the vesicles budding from the cell. And these are just increasingly high magnifications of this process. So the conclusions at this, after this first part of the talk is that protein budding is induced by high-order oligomerization and plasma membrane binding, but that targeting these same cargo proteins to the endosome membrane, in our hands at least, does not induce efficient protein budding. And the main takeaway here is the plasma membrane needs to be considered as a major site of protein budding, and not just any kind of protein, but exosomal proteins as well. And this is important, particularly for future translational studies on exosomes. If the goal of the research being funded by NIH and other organizations is to go beyond the understanding of the basic biology and actually to translate that information to making vesicles that we can load with cargoes that we want and target to cells and tissues that we want, we need to know the rules that the cell uses for targeting proteins to exosomes and then copy those rules. And the bottom line is this: if you have a protein that you want to target into exosome for some therapeutic purpose, targeting it to the endosome membrane is not actually going to get you there very efficiently, whereas targeting it to the plasma membrane will do a much better job. Okay, I want to add in another layer of into the talk, which is that of virus budding. Shown here is a cartoon of retrovirus life cycle, where you have the virus particle on the far left, upper left, coming to a cell, engaging with its receptors and co-receptors, fusing the membrane between the plasma, between the cell and the virus. You have an uncoating process where the capsid gets out into the cytoplasm. It translocates to the nucleus where there's reverse transcription and then integration of the proviral DNA. And then from that provirus, expression of RNAs, translation of proteins, assembly of a new particle, and budding of that particle from the plasma membrane. This is the basic, and then once it's budded, you have the maturation step, and then you have a fully functional retrovirus ready to infect another cell. Now, in studies by James Hildreth and many others, it's become clear that HIV particles bear a great number of similarities to exosomes. They have the same basic topology. They have almost exactly the same size. They have the same host lipids, the same host proteins, and studies on the carbohydrate profile of the glycoproteins and glycolipids show that they have very similar carbohydrate signatures as well on their extracellular domains. Now, this led James and I a number of years ago to explore this idea that retrovirus budding, particularly HIV budding, is a form of exosome biogenesis. And clearly, in addition to the similarities in the finished particles that are released out from the cell, there's parallels in the cell biology, where you have a lot of evidence for endosomal budding, a lot of evidence for plasma membrane budding in both cases. And this led us to this Trojan exosome hypothesis that Chad mentioned earlier, and which is just diagrammed right here in these two slides. That when retroviruses infect the cell, their capsid proteins and other structural proteins are viewed by the cell and treated by the cell essentially as exosomal cargo proteins. They're targeted to sites of exosome biogenesis, and they use the exosome biogenesis pathways to make it the infectious virion. Now, some of the supporting evidence for this comes from isolated studies of the viral GAG protein, not just the virus itself. So just an overview of retrovirus structure. The proviral genome consists of LTRs flanked by several open reading frames that encode, for all retroviruses, GAG, a GAG pol fusion protein. The pol encodes the reverse transcriptase integrase protease functions. And then the envelope protein, a separate protein that's made from the spliced transcript off the proviral genome. Now, a large number of studies over the years have established that it's not the envelope protein that mediates the budding of cells for retroviruses, but rather it's the GAG protein that mediates the budding. So to study virus budding, many people in the retrovirology field study the budding of GAG protein, and that's where we initially put our focus as well. One of the things we observed right off the bat was that if you look at the expression of GAG in human T cells, you find that it also goes to these cap domains of the cell surface, where it co-localizes with CD81, CD63, shown here. Again, consistent with the notion that GAG, HIV GAG, is treated as a cargo of the exosome biogenesis pathway. Now, if one takes another view of retroviruses, as do the GAG proteins actually have these features that we've found to target proteins to sites of vesicle budding and into exosomes? It turns out they do. All GAG proteins have some type of a plasma membrane anchor. It can be a myristylation signal. It can be a PIP2 binding domain. It can be both. In the case of HIV, it actually has both a PIP2 binding domain and a myristylation site. And also, GAG proteins have multiple oligomerization domains, which is what you need in order to form a high-order oligomerization. Another way of thinking about that is that they form controlled aggregates. So in the case of HIV GAG, you have a matrix region that has a trimerization domain, a capsid region that is a hexamerization domain, nucleocapsid, which has some dimerization activity. Of course, the GAG protein is named by the products that are generated by the action of the viral protease. So if you just express GAG, you express this entire protein, the whole 55 kilodalton protein, matrix capsid, NC, as well as the spacer peptides in the P6 domain. All of that is the GAG P55 protein. And we're mostly going to be just concerned with the budding of that protein, not how it gets cleaved by protease or what all the different subparts of the GAG protein are for. Anyway, so the GAG proteins all look like cargoes. They all behave like cargoes because when you express them in mammalian cells, they all go to sites of vesicle budding, and they come out of the cell in association with exosomal markers. I'm not showing the budding in this slide, just the intracellular localization. And the point is that they all co-localize with exosomal cargo. So here's the budding of equine infectious anemia virus protein, HTLV1 protein, murine leukemia virus protein, Rous sarcoma virus, Mason Pfizer monkey virus, the endogenous HERV-K that we all express, all have in our genomes and all express. They all go to sites of budding and bud from cells in exosomes. And this is not true for a class of viruses that are related to retroviruses but different. Simian foamy virus, GAG-like protein, does not bud from cells, does not localize to sites of budding, does not come out of the cell in exosomes. So it's a general feature. It's not a specific thing for HIV. It's true of all retroviruses. Cells treat them as GAG proteins. And you can see this at the ultrastructural level as well. Here's a cell infected with HIV and immunostained with antibodies to CD81. And you can see here the immunogold label for CD81 at this budding profile. And we know it's a budding profile because it has that electron-dense GAG oligomer. And that's true also in these images below. And not just that the CD81 is on the virus particle, but it's in the vicinity of the budding. And it's demonstrated here even in a more nascent budding profile as well. If one collects the vesicles from cells that are expressing GAG and expressing a lipid marker of exosomes, here rhodamine labeled PE, one can simply dry them onto the glass and ask the question of whether or not they're co-localizing at the same vesicles. So to the right panel, each of these is a vesicle dried down onto a glass slide and imaged to detect the rhodamine stain. And then we took a picture to look at the GFP stain, and GAG GFP is present only in vesicles that are also labeled with the exosomal marker. Now, that data suggests that when HIV buds or HIV GAG buds, it's budding from exosomes. If that's true, then if we just artificially cross-link any protein and get it into the exosome pathway in cells that are also expressing GAG or infected with HIV, it should target those onto virus-like particles and target them onto virions. So here I'm just showing you again what happens when you cap CD43 in a T cell. If this T cell is expressing at the same time a DS red form tag of GAG, you find they co-localize at the same domains of the plasma membrane right out here. Okay, and if you collect the exosomes from these cells and again dry them on glass and visualize them by fluorescence microscopy, you can see the vesicles, all these vesicles that are labeled with red labeled GAG in circles. And these are the ones that are also labeled with the cross-linked CD43. So this is CD43 that's been stained with a primary antibody, cross-linked with a secondary antibody, and then the vesicles collected from the culture supernatant dried down, and you can find CD43 now on vesicles. They are also containing GAG. You can do this in cells that are infected with HIV and get the same sort of result. To the left panel here is HIV budding from T cells. These T cells were stained with antibodies to CD43, but we did not cap the CD43. And one can find some CD43 in the area. There's one gold particle, I believe, right down here, but they're not enriched in this area. The CD43 is everywhere, so it's not excluded from sites of budding, but it's not enriched in sites of budding. But if now we cap CD43 and we look at the brain density with the immunogold staining, it's much higher. We find evidence for these cross-linked CD43 complexes on the HIV particle. And the increase is roughly 20-fold in the CD43 concentration on the HIV particles. Now, one thing I also mentioned early on was that the exosome protein sorting pathway is involved in polarization of cells, particularly we would refer to as amoeboid polarization, cancer cells, but also in the polarization of leukocytes, which also form an anterior-posterior polarity. And when cells do adopt this type of polarity, they have a uropod at their posterior pole. Here's the uropod at the back of the cell. There are certain proteins that are known upon polarization to go to this domain. CD43, interestingly enough, is one of these. When it does so, what you also find are the exosomal cargos going to this domain. And the reason I'm bringing this up in this talk about HIV is that if you now look at where HIV GAG goes in cells that are polarized in this manner. So here's a human neutrophil-like cell line, HL60. We modified to express HIV GAG GFP. And then we put it on a plate and gave it a chemoattractant. The chemoattractant is coming out of this pipette. You can see this neutrophil here is migrating rapidly towards this pipette tip. This is the anterior end. The posterior pole, this uropod extension, is lighting up brightly for HIV GAG. And if you look at these cells at the ultrastructural level, what you see, again, here's the anterior end of these cells, anterior pole of these cells. Here's the posterior pole of these cells. And from the posterior pole of the cells, you see exosomes budding. You see virus-like particles budding. offer this. And so again, there's this correlation that shows up again and again and again. Whatever you find about exosome biogenesis or exosomal protein trafficking, that pretty much holds true also for the trafficking of HIV GAG, into the budding of GAG virus-like particles, and budding for HIV virus particles in a regular infection as well. Now, this notion that James and I had a long time ago about HIV being a Trojan exosome, retroviruses being a Trojan exosome, actually also has some relevance for how we think about the evolution of retroviruses. For many years, retrovirus field has believed that retroviral evolution was initiated primarily by the transduction of an envelope type gene, LTR retrotransposons, which are present in organisms like yeast that don't have any envelope virus budding. They have a GAG-like protein, a GAG pole-like fusion protein linked by long terminal repeats. And of course, retroviruses have this other gene, the ENV gene. So obviously, it's a major event in retroviral evolution in the acquisition of envelope gene. But for that to be the defining transition between a retrovirus and a retrotransposon is asking an awful lot of that ENV gene. That ENV gene has to, get the capsid structural protein and the genomic RNA into the membrane, gives it a membrane. It has to mediate its budding from the cells. It has to mediate the binding to other cells, and it has to mediate the fusing with other cells. And if it doesn't do all those four things, it really doesn't give any selective advantage to the transmission of this retro element. And an exosomal view of this evolutionary process is a little bit simpler. In an organism that already is doing exosome biogenesis and intracellular exosome trafficking, you don't need all of those events. In fact, you don't even need the transduction of an envelope gene. All you really need is a glycine substitution mutation at position 2 of whatever this main structural protein is, the GAG-like protein. Because if that becomes a glycine, now it's subject to being myristylated. That will traffic the structural protein to the membrane to sites of vesicle budding. And given that these structural proteins bind the genomic RNA, and will bring the RNA into the vesicles, if these vesicles, these exosomes being released, are transmitting from cell to cell, gives the ability of the retro element to get into the cytoplasm of a neighboring cell, here, and then complete a pathway of cytoplasmic and nuclear replication. So it initiates in one cell over here with the expression of the RNAs and the proteins, leads to the budding of these nascent protovirions, and their release out into the cytoplasm, and then their entry and their reinfection of another cell. So this is, makes possible the transition of a transposable element into a virus just by a single point mutation in one protein. So to summarize this part of the talk up, I'd like to say that the evidence is pretty clear that retroviral GAG proteins are targeted to sites of exosome biogenesis and secreted from the cell and exosomes. And the HIV virus itself is budding from cells together with exosomal markers, and that high order oligomerization is sufficient to target plasma membrane proteins to HIV particles. Now, I think this is a pretty convincing set of data, but really when you look at the HIV field, you don't see a lot of people modeling it as an exosome. And this is reflected in the views of a lot of people, and including NIH, and this is just a really sort of sharp reminder of this. This year, NIH put out an RFA, the title of which is to explore the role of exosomes in HIV pathogenesis, and in the instructions for the application, it says specifically the application will not be reviewed the study the use of the exosome biogenesis pathway for HIV formation. So it's hard to get traction with this idea in the HIV community in general and with NIH funding. And why is this? After all, I've told you that the GAG proteins look for all the world like an exosomal cargo. They have a membrane anchor. They have multiple oligomerization domains. They form very large organized aggregates of about 50 megadaltons or larger. And all you need for that is matrix capsid and nucleocapsid. You don't need any other part of the GAG protein for that. And this gets to be part of the problem with the adoption of this idea, because in the HIV field, it's been known for many, many years now. And this was work initiated by Heinrich Gottlinger back in Bill Hazeltine's lab. So going back to a paper published in 1991 in PNAS, if you look at the budding of HIV from many adherent cell types, you see results that are kind of shown here. To the left panel are cell lysates of a full HIV provirus or virus, ML4.3. And it's a cell-associated amount of the GAG protein. And now this is the vesicle amount released in virions. And we're looking here at the breakdown products of GAG. Starts off as P55, but then gets cut down by the viral protease to the 24 kilodaltons. And our antibodies are specific for that 24 kilodalton capsid piece of the protein. Anyways, okay, so you infect cells with virus. You can see the GAG protein in the cells, and you can see the GAG protein in the virions. Not surprising, everything is fine. What Gottlinger and Hazeltine showed was that if you had a mutation, this mutation called L1-PER, and this is a mutation that stops the protein right after the second spacer peptide. So it prevents the synthesis of the P6 part of GAG. You can see it expressed in the cell. There's the band. And what you don't see is a corresponding band in the virions. You actually do see some incompletely processed GAG proteins in the virions, but you don't see very much of them. And so based on this data, it was concluded that the P6 domain was necessary for budding and was actually mediating the budding. So this actually runs quite counter to our argument that all you need is the upstream regions of the oligomerization domains and the membrane binding domains of GAG in order to bud from cells. And furthermore, people in the HIV field, Wes Sundquist and others, have done some beautiful work actually defining this at this P6 role at an atomic level, and a biochemical level. So it turns out that in the P6 domain, there are sequences, particularly the PTAP sequence, that binds to a component of the escort-1 complex, that's DSG-101. And these escort machineries are thought to catalyze virus budding and vesicle budding in general, in the absence of viral infections. And I don't want to get into all the data involved in this question, but I just want to point out that even though it is quite clear that loss of the P6 domain causes a defect in budding, and that there's good reasons to believe that it has something to do with the TSG-101 interaction with the P6 domain, people kind of stopped doing experiments at that point that would question that model. And given that we were finding, working from a different model, we followed on and did some experiments to push this idea and question this idea of whether the P6 domain really is important for budding. And I'm going to summarize some of these right here. It's pretty straightforward stuff. Here's your GAG protein. It's 55 kilodaltons, and again, it consists of sequences that can be thought of as the composition of the matrix capsid, SP1, NC, SP2, and P6 domains, all as a single protein. If you express that in cells, and here now we're in 293 T cells, what you'll find is that you have cell-associated amount of protein, and the protein buds really, really well. If you make the kind of mutation that was reported by Gottlinger many years ago, where you simply stop expression of GAG, or stop codon right at the beginning of P6 domain, and we'll call this GAG P49, you get very little budding. So you can see there's very little released in the exosome fraction, and if you do this multiple trials, you can quantify it and show it's more than a 5-fold decrease in budding. But here's the curious thing. After exactly why we went to do these experiments, I don't want to get into, but we did them. If one deletes further into GAG, so now we go to GAG P47, that's this protein down here. Okay, and what you can see is that it not only does not have the P6 domain, but it also doesn't have that sp2 domain. So this consists of matrix capsid sp1nz. If you look at its budding, it buds great. And the budding is quantified here from multiple trials. It's as good as wild type. So it seems that the budding defect caused by loss of P6 really wasn't the loss of P6. It was the exposure of a new C-terminus, this sp2 at the C-terminus. And to test that a little bit further, we took the full-length GAG protein and we simply added those 16 amino acids of the sp2 domain onto the C-terminus of it and asked whether it could bud or not. And the results shown here on the bottom in panel C, and you can see in the immunoblot and the exome fraction, wild-type GAG buds very well, and then the mutant GAG sp2 protein doesn't bud hardly at all. If you quantify it, it's basically a 20-fold reduction in budding, this GAG sp2. So putting the sp2 peptide at the extreme C-terminus of the protein, HIV GAG, whether you're adding it to the full-length GAG or you're getting there by deleting P6, generates a protein that cannot any longer bud from cells for some reason. Now, it turns out it's a transferable property. So if you now take this 16 amino acids from the sp2 domain and you add it onto other retroviral GAG proteins, they won't bud themselves anymore. And I'll just draw your attention here to the results with HTLV1. In this panel here, this is the cell-associated or the wild-type protein. Here's the budding of HTLV1 GAG proteins in this lane. You express the GAG with the sp2 domain. It expresses quite well in the cells. It buds very poorly from cells. This is not restricted to retroviral GAG proteins. You can put it at the C-terminus of CD63, and it blocks and inhibits the budding of it as well. We even have completely synthetic cargos that bud quite well. Here's one, just an acylation domain with a leucine zipper and a tetrameric protein. It buds from cells pretty well. But if we add this sp2 domain to the C-terminus, it doesn't bud from cells anymore. So this inhibitory budding signal is sort of there cryptically in the GAG protein. It's exposed by certain mutations, lab-generated mutations. But it's telling us something interesting about budding because it's able to stop it. Now, working from the GAG protein with the sp2 at the C-terminus, we did a point mutation scan across, an alanine scan mutation across that peptide, and identified residues that, when mutated, eliminated its ability to block budding. So here's the budding of wild-type GAG. Here's the GAG sp2, doesn't bud very well. And here's the budding of all the various different mutants that we generated. And this allowed us to identify functional amino acids in this inhibitory signal. And one of the things we wanted to test was whether this could explain the budding of the virus. It's important in the HIV field to take whatever you find for the GAG protein and then replicate it in the context of the whole virus, which is large. I'm not showing you the whole viral sequence here, but here's what we did. We generated mutations that allowed us to do an epistasis analysis of HIV budding. So these have the P6 element that blocks budding. And then we've added in other mutations in the sp2 domain, either so we can look at the budding defect caused by the mutant virus, and then whether now mutating these inhibitory budding signal amino acids could restore the budding. The bottom line is they did. They don't give us proper processing, but when we combine these IBS mutations along with the P6L1 term mutation, we get back normal budding. So my conclusion here is that the precise deletion of P6 domain exposes a cryptic inhibitory budding signal in HIV, that deletion or point mutations in this signal restore budding of P6 deficient GAG and HIV, give us a much more sophisticated view of what P6 is doing, why it's interacting with escort machinery, and how that relates to budding of virus and to processing of virus by the viral protease. So giving us new insights into HIV biology and posing new challenges for us and for the field. And what I'd like to do to finish up here is just to summarize future directions in this field. Obviously, we need to make a lot more progress to elucidate molecular mechanisms of protein budding. We need to explore their contribution to virus budding, especially for retroviruses like HIV, and then translate these advances to make synthetic exosomes that can deliver therapeutics to specific cells and tissues. And with that, I'll conclude my talk and remind you all, please join us in Florida, October 16th and 20th for ASCMV 2015. And with that, I will conclude my talk. And thank you very much for your attention.
Great, thank you for that terrific presentation, Dr. Gold. I am excited that your exosome Trojan hypothesis is starting to gain some traction. Before we get started on our question and answer session, I would like to remind our audience that you can submit questions by typing them in the Q&A box at the lower left of the presentation window. Throughout the presentation, we have received several good questions. Our first one is in regards to inhibiting exosome formation by blocking N-SMase enzymes. For example, does that also block HIV budding?
Well, yeah, there's reports. The original report by Trajkovic et al. In Science reported that it does not block that. So there are, it's also clear that some exosomal cargoes are not blocked by inhibiting N-SMase enzymes as well. So
it might be. Great, thank you for that answer, Dr. Gold. Our second question, and a theme throughout our questions that came in, are what methods are you able to separate exosomes from virus particles?
yeah, the standard method for separating them from virus particles is based on density. And that's allowed because the virus particles usually incorporate very large amounts of their major capsid protein into the vesicles, which changes the density. So in my opinion, those still are exosomes at a different density, but you can, for let's say from HIV-infected cells, you can separate exosomes that have the fully formed capsid inside of them from the exosomes that do not have fully formed capsid inside of them, just on a density centrifugation gradient.
Great, thanks a lot for that. Unfortunately, we're out of time today. I want to thank the audience for their interesting questions and participation in today's event. I would also like to thank our sponsor, Beckman Coulter, and of course, Dr. Stephen Gold for making today's educational webcast possible. This presentation will also be available on demand viewing through beckmancoulter.com. Thanks again. Goodbye.